Topic is genomics. Let's see about genomics. What is genomics? The genome is the entire set of genetic instructions found in a cell. In humans, the genome consists of 23 pairs of chromosomes found in the nucleus as well as a small chromosome found in the cell's mitochondria. Each set of 23 chromosomes contains approximately 3.1 billion bases of DNA sequences. Genomic-based healthcare involves use of this genomic information both an individual as part of the clinical decisions. Example for diagnostic or therapeutic decision making and consideration of implications of that use. History of genomics. In 1977, Frederick Sanger developed a sequencing technique for DNA to sequence the first complete genome called X174 virus, which opened the doorway to the possibilities in the field of genomics. In 1980, he won the Nobel Prize with Wally Goldberg and Paul Berg for pioneering DNA sequencing methods. Difference between genomic and genetics. Genetic is the study of heredity. Genomic is the study of the entirety of an organism's genes. Gene refers to a specific sequence of DNA on a single chromosome. Genome refers to an organism's entire makeup. Genetics involves the study of functions and composition of the single gene. Genomics addresses all the genes and their interrelationship. Subfields of genomics. The different research areas of genomics can be as follows: structural genomics, functional genomics, comparative genomics. Structural genomics. Construction of genomic sequence data, gene discovery and localization, construction of gene maps. Functional genomics. Biological function of genes, regulation, products, plant development studies. Comparative genomics. Compares gene sequence to indicate functional or relationships. And is the genomic sequence process. Goals of genomics. The goal of the Human Genomic Project was the complete comprehension and mapping of all genes of humans in the DNA sequence. All our genes collectively or genomics. The main aim was to determine the number and sequence of all base pair in the human genome. Why to sequence genomics? Sequencing genomics helps understand how the genomics as well world and how the genes work together to direct the growth, development and maintenance of an entire organism. The genome sequence will represent the valuable shortcuts, thus helping to find genes much more easily and quickly. Technical foundations. The technical foundation of genomics involves construction of genomic and DNA libraries, DNA identification, restriction, enzyme mapping, DNA sequencing, PCR amplification. Steps in genomic sequencing. Break genome into smaller fragments. Sequence those smaller pieces. Piece the sequence of the short fragments together. Genome sequencing approaches. Hierarchical shotgun sequencing and whole genome shotgun sequencing. Human Genomic Project. The Human Genomic Project is an international scientific research project with the goal of determining the sequence of chemical base pair which make up human DNA and of identifying and mapping all of the genes of the human genome from both a physical and a functional standpoint. HGP was formally founded in 1990 by the US Department of Engineering, Energy and the National Institute of Health and was completed on 14 April 2003. These are the basic goals of HGP. Benefits and applications. It is also useful in the field of agriculture to study evolution through mutation, advantages and disadvantages. Thank you.